Alright, we like to start with a monohybrid cross to get our Punnett square practice. Remember, mono meaning one, so we're looking at a one or single trait cross. And we have the genotypes of the parents. The parents following genotype, we have homozygous, recessive, cross with heterozygous. So remember, one letter, we have one gene, uppercase, dominant, lowercase, recessive. The gene comes in two different forms of a gene we call alleles. So here we have the cross, and like we've talked about before, we start by taking one parent, and this is arbitrary, but I recommend doing the same system each time so that you have a routine, a procedure. Take the letters on the left-hand side and split them down on the left-hand side. The right ones, split them across the top. So remember, the parent is diploid, and then during the production of sperm and egg gametes by meiosis, we're going to basically cut this in half and make haploid cells. So, for example, this could be the sperm. If this is the male and if this is the female, then these represent egg. The egg and sperm, the gametes have half the genetic material, so that when they fuse during fertilization, we restore the diploid number. That's the power of a Punnett square. Not only does it help you to predict probability, but it also helps us emphasize the biology behind it as well and understand basic principles of genetics. So these are the possible gametes that the male can form, lowercase a, recessive allele, and the female can form eggs with a dominant or recessive allele. Now we want to look at what are the possible combinations we could get. So the Punnett square itself, each box represents the potential offspring. So we bring the capital down. Now, is it okay to write little a, capital A? Yeah, it means the same thing, but by convention we always put the capital first. So capital A, lowercase a, little a, bring this capital down, capital A, lower a, lower a. So when we look at genotypic frequencies, we see that we have 2 boxes out of 4, or 1/2, 0.5 chance that the parents are going to have a offspring that is homozygous recessive, and then 1/2 chance that they're going to have heterozygous. Again, our fractions should add up to 1, decimals should add up to 1, percent should add up to 100. It's a good way to check. 0.5 gives us 1.0. All alleles are accounted for. That's the basic steps, the simplest genetic cross, monohybrid.